So welcome everyone to the functional analysis panel. I'm thrilled to be speaking to three experts today about how variants are functionally classified. I'm just going to quickly share my screen. We are joined by Dr. Steve Traynelis, who is a professor in the Department of Pharmacology and Chemical Biology at Emory University School of Medicine. Dr. Ian Coombs, who's an experimental officer and lecturer in the Department of Pharmacology at the University College London, and Dr. Lonnie Wollmuth, who's a professor in the Department of Neurobiology and Behavior at Stony Brook University. I'm just going to actually get started with a very brief introduction on what variants are and what functional classification means for the families in the audience, and then we'll get into some specific questions for our panelists. So first, just an overview of genes and DNA. If families have genetic reports available or a variant handy, feel free to follow along, but if not, no worries, this will still be understandable without that. So genes within DNA encode proteins, and the proteins are what carry out all the functions necessary for life. So genes provide those fundamental instructions using a 4-letter alphabet code. So as you can see here, A, T, G, and C. And then machinery in the cell reads that code, translates those instructions into this intermediate step called mRNA or messenger RNA, and then into amino acids, which are the building blocks of a protein. So each 3-letter word codes for a single amino acid, and then a chain of amino acids strung together makes a protein, and that protein carries out a particular function in the body. So each GRI gene, as you can see on the right-hand side here, encodes a different receptor. GRIN genes encode the building blocks of NMDA receptors. RIA genes encode proteins that make up AMPA receptors, and so on, as shown here on the slide. Now, spelling errors can happen in these genetic instructions, and that's what we refer to as genetic variants or mutations. I'm going to be using those terms interchangeably here. And that can actually lead to changes in the mRNA and ultimately the protein. So you can see here that that amino acid has been switched out. And these typos can happen either spontaneously, de novo is another word for that, or they can be inherited and can affect the structure and ultimately how the protein behaves in the body, and that can lead to symptoms. A wild type gene, on the other hand, is just a gene that does not have a variant. So this slide might look familiar to some of you, but just showing again the different types of variants. So variants can arise from single letter substitutions in the DNA that change one single amino acid in the protein, and these are called missense variants or mutations. They can also create a premature stop signal. So the machinery in the cell is reading those DNA instructions, and then all of a sudden it receives a signal to stop. And what that does is it leads to a shortened or truncated protein. And we call these nonsense or truncating variants. Another example is the addition or removal of DNA building blocks. So that's in that 4-letter code. We call these nucleotides. And if they're not a multiple of 3, it completely shifts how the instructions are read by the cell machinery and scrambles the rest of the code. And so typically that results in a protein that is completely non-functional or shortened, and we call these frame shift variants. In addition to the variants that I'm showing on the slide here, there can also be larger scale deletions or duplications. So, they're not limited just to the ones that I'm showing here. So GRI genes encode a class of receptors, which are a type of protein, and these are called ionotropic glutamate receptors. They play really important roles in synaptic connections and communications between neurons. So here you can see 2 neurons communicating. So they're necessary to transmit electrical signals between neurons, and that's how they speak to one another. And that makes biological functions like cognition, sensory processing, and movement possible. So these receptors are specifically activated by the neurotransmitter. So this is present in the body called glutamate. So glutamate sends a signal, and the ionotropic glutamate receptors receive that signal and activate that receptor or turn it After activation, the receptors allow ions to flow into the neuron, and that triggers a chain of events in the neuron that actually leads to those biological functions. So variants in GRI genes can have different effects on the glutamate receptors. The resulting functional effect really depends on the particular variant. So some dial up the volume of a receptor and can kind of amplify neuron-to-neuron communication, while others dial down the volume and dampen neuron-to-neuron communication. So variants with these effects are referred to as gain of function or loss of function, respectively. One thing that's really important to understand is that these effects happen in the body, and they can lead to similar symptoms. This is something I was talking about with the census data as well in the last session, is that we can't tell whether a variant is gain of function or loss of function simply based on the symptoms that someone experiences or their physical characteristics or anything else. So a lab technique called electrophysiology, this is actually the technique that I use primarily during my PhD. This is commonly used to characterize a variant as gain of function or loss of function. So researchers can record electrical activity and ion flow across the receptors to see how they're communicating in cells in a dish. And this allows them to test whether the amount of ion flow is, amplified or dampened, or, if something else is going on as a result of that variant. And once the functional effect of a variant is known, researchers can also test which drugs may kind of compensate for the effect of the variant. So for example, there are different drugs that can increase or decrease activity of these glutamate receptors depending on the type of variant. For individuals with GRIN disorders, so variants in GRIN1, GRIN2A, GRIN2B, or GRIN2D, I just wanted to highlight this, that families can check the GRIN portal. This was developed by Drs. Lemke, Traynelis, Benke, and Lal. And so if you go to the GRIN portal and if you click on variant classification, select the gene, and then type in a particular variant, then you can see the functional information. So this notation that you're seeing on the slide is typically what the information looks like on a genetic report. So you'll see C dot followed by numbers and letters, and a P dot followed by numbers and letters. The C dot basically just describes the variant at the DNA level. So what is happening in the DNA? And the P dot describes the variant in terms of what's happening in the protein. So they're just two different ways of describing the same variant. And here, as an example, I just chose a variant where glycine is replaced with serine. So that's the amino acid change that actually happened as a result of the variant in GRIN2B. And you can see here that this particular variant was classified as likely loss of function by considering multiple parameters, as you can see here, that affect NMDA receptor function in different ways. So that's all I have for an introduction. So now we're going to move into the question period. And I see that Steve is here now as well, so hi. So I'm going to start off asking the three of you to kind of talk really broadly about why it's helpful for families to know whether a variant is gain of function or loss of function. So for example, does this change the medications an individual could take, what clinical trials they could participate in? Like, why does it matter? So we'll start off maybe with Lonnie, and then we'll go to Ian, and then Steve.
Thanks, Quinn, for that very nice introduction to the general issue. Yeah, the idea is that understanding loss and gain of function, being able to define a variant if it's loss or gain of function, really is one major strategy we think about of how to target them in the clinic. Again, a loss of function variant, you could envision that if you enhanced its activity, using certain compounds, which are called positive allosteric modulators, or PAMs, that you might be able to at least reverse some of the clinical deficits. Similarly, with a gain of function, you could use compounds such as negative allosteric modulators, or NAMs, that might tone down the activity of this hyperactive receptor. So being able to define the variant as loss or gain of function is really sort of a major first step to defining, what potential clinical strategy might be useful to use to target them.
That's great. Thank you. Ian?
Yeah, I mean, what can I add to that? That was very nicely put. Yeah, in terms of the need to understand it in a way is slightly dependent as well on what drugs are actually available. So it's clear that if you have a gain of function variant, it makes sense that you would use some kind of inhibitor, a negative allosteric modulator, and a PAM for loss of function. But we still don't necessarily have that many tools that are able to directly counteract these variants, and so it's not at the moment enough to know loss or gain of function for many of the genes, because we can't use that information to use a precision therapy, because for example, if you have a GRI loss of function, even though hundreds of I mean, possibly that's a slight exaggeration, but tens and tens of compounds that are able to act as AMPA receptor PAMs have been developed, and they work in an experiment in a dish, but none of them have made it into the clinic. So knowing you have a loss of function GRIA is just at the moment is important information, because you definitely don't want to use a negative allosteric modulator. But we need to keep on driving forward our understanding of variants, but also driving forward our ability to target them once we have that key piece of information.
Great. Steve, do you have anything to add to that?
Just a couple of points. I mean, very well said by Lonnie and Ian. I think they hit the nail on the head. The only couple of points I think would be worth mentioning are, gain and loss are, very old. It's very old terminology, as Lonnie nicely said it. It's just the first step, but you've got to take the first step to do the next step. And in determination of gain or loss, at least in the NMDA, the GRIN world, it involves at a minimum multiple assays, and so you already have a baseline of half a dozen parameters. And as the field and researchers move forward and drug development programs gain momentum, these other parameters will be examined and almost certainly at some point used to differentiate patients in clinical trials. And so, it's a hopeful view, but gain or loss sort of puts the variant in play for future consideration. And just to Ian's point, there are many hundreds of compounds, as he nicely said, for AMPA, but the AMPA receptors are really, really complex. I think it's a harder problem than an NMDA, because they, as will probably come up in the conversation, and Ian's an expert in this, and Lonnie's worked on it too, there are many, many accessory subunits. And so gain or loss, defining gain or loss for an AMPA receptor is going to be a significant challenge for the field. But if you don't take the first step, you're not even, it's not even in play, and you don't even know as things move forward where to go. And then just one other point that's really important, it's derivative of what was mentioned, but not only do you want to pick the right patients for the clinical trial, but you want to help the people designing the clinical trial stratify the patient populations. These are really unusual trials. For example, the Radiperdil trial going on now is a 100-patient pivotal phase 3 single trial that'll lead to either approval or rejection of the medication. Any other drug has to do two phase 3 trials, usually 1,000 patients or something. And so the trials are really small, and what that means is that any undesired or unexpected heterogeneity in the patient population that you're trying to treat, if you have a precision medicine and your in vitro and preclinical work tells you it's going to work a certain way, but you broaden the trial too much, a few patients can sink that trial. And so the more you know upfront about the patients, the better position the clinicians who are actually, running the power statistics and the math and thinking through, what are the parameters we need to hit to get a statistically significant result, that, the better job they can do. The more patients that they have data from, whether even if it's a lost patient and it's a gain trial, the larger the universe in numbers, the better job they can do in that trial design.
So actually, both of you, or all of you, briefly touched on the next question I was going to ask was, is it always as simple as gain of function or loss of function? It seems like the answer is no. So what specifically, like if it's not that simple, then what specifically could be feasibly tested in a lab to understand a variant better before, someone enters a clinical trial, let's say? Like what can we feasibly do beyond gain of function and loss of function? We can start maybe with Steve, since you were just talking about.
Sure. So, in trying to figure out how to call gain or loss of function, there's an awful lot of work involved, and there'll be a ton of work involved for the AMPAs. Just as I mentioned, they're so complicated, and I'm sure it would be, Ian, be great for you to amplify that. And so there's an awful lot of background recording, characterization, discussion amongst, the experts about, what parameters are we going to use to make this determination? What parameters would we like to know in a perfect world down the road that would help us? And what parameters, pragmatically is a bridge too far? It's just too much time, too much money, to the point where if we tried to do it, we would fall short on everybody else. And so, beyond gain or loss, what the answer to your question is, what's the utility of other things? It's more functional characterization that has been discussed by the field, accepted by the field, and then is pursued in a unified way so it can be compared, whether it's done in London, whether it's done in New York, Atlanta, Tokyo, Paris, wherever.
Thank you. Ian, do you have anything to add to that? And then Lonnie?
Yeah, in terms of the complexity, I guess this comes down to resources. I mean, in general, expression system experiments are, on the scale of things, relatively cheap, certainly a lot cheaper than making a mouse and handling animal models. But nonetheless, you need somebody to do the experiment. And in terms of the AMPA receptors, they work when there's only one subunit there that's all the same, unlike NMDA, where you need GluN1 and then most likely some GluN2 subunits as well. AMPA receptors, they can be only one subunit, or they can be heteromerous, or they can be triheteromerous. And then you have the auxiliary subunits kicking in as well with the TARPs. I know Derek spoke about this earlier, and there are various other auxiliary subunits as well. And more variants are cropping up as well. And I think, yeah, that is a limitation, certainly with the AMPA receptor stuff, is that you don't have the time to put these variants in every single combination. And so you end up looking at a very few standardized combinations, and you hope that reflects broadly what the receptors are actually going to do in the brain. Typically, I would be generally surprised if an AMPA receptor that looked gain of function when it was on its own and looked gain of function whether it was one with one auxiliary subunit ever looked a different way with another auxiliary subunit. But until you kind of try them all, you don't know. So it may be the case that we need to take a few example variants and prove with, in 10 different combinations of heteromers and auxiliary subunits that they pretty much look the same in all of them. Because if you try and do those 10 experiments on every variant you've got, I mean, we can't keep up with just doing one combination of variants at the moment. So there are, the main resource is time. You need people to do the experiments. And that is, it's always hard getting funding. Yeah, I guess that's where I'd leave that.
And just to clarify for everyone in the audience, when you say TARPs and auxiliary subunits, these are basically like helper proteins that, keep the AMPA receptors where they're supposed to be, and kind of change their function. Is that right?
Yeah, that's right. Yes. So you can, in our experimental systems, you can make an AMPA receptor that's only an AMPA receptor work, and you can look at its properties. But in terms of what you find in the brain, they will almost always have some combination of auxiliary proteins with them. Probably the majority of receptors probably even have as many auxiliary subunits as they have subunits, so probably 4. Now that seems to be what comes out of most of the native structural work that's going on at the moment. But yeah,
right. So what's happening in the dish is like a little bit different than what's happening in the body, of
Yeah,
course.
well, we try. I mean, our standard conditions actually include auxiliary subunits, but
Okay.
they only include one kind of auxiliary subunit, and they only include one kind of AMPA receptor subunit. So even that is like it's a compromise. You know, ideally we would know how it behaved in every combination, because for one variant somewhere there's going to be one that behaves differently with an auxiliary subunit in a way you don't quite predict. But it's unlikely that we would necessarily find that because of the man hours it takes to replicate the experiment 10 different ways. So
Right. All about balance. Lonnie, do you have anything to add to that?
Yeah, I was just going to say, yeah, I mean, Steve's making it sound like AMP is super complicated and NMDA is fairly straightforward. I mean, I think, and in ways that's true because there's fewer interacting proteins that the NMDA receptor deals with. On the other hand, one of the features that make the NMDA receptor so important in brain function is its really complex way of working. And so trying to define, in a simple way, whether it's loss or gain of function is sometimes challenging because there are so many features of the NMDA receptor that make it not only carry current, but in being able to be altered in many ways. And so this adds to the complexity of the NMDA receptor. And when you characterize it or loss or function, you have to characterize many properties. The Center for Functional Evaluation of Rare Variants, they characterize 6 properties right now. And again, that takes time and effort. And the related issue is that there are a lot of NMDA receptor variants. There's over 1,000 of them now distributed throughout the protein. So it's a non-trivial task to characterize these proteins. But
Yeah,
it's a
and maybe
critical
I'll
step.
for
Critical
sure.
step.
And maybe just we will leave time at the end for audience questions, but there's one in the chat that particularly touches on what you were just saying, and that is where are we at in terms of characterizing variants? Is it realistic to have all of the variants characterized?
Steve can answer that question, but if you GRIN portal, I think is really an invaluable resource both for clinical information, but also that's where the information from the Center for Functional Evaluation of Rare Variants goes. And families can go and look at that site and see if the variant has been characterized or not. So, and a lot of them have been characterized. I think 415. I just looked at GRIN portal right now.
Yeah. So it's a good question in the chat. Is it reasonable and where are we? And so, Lonnie again, cast the the view correctly. There's well over 1,000. I predict we'll top out around 2,000. I think the sodium channel with Dravet syndrome and GRINs will sort of asymptote to each other. They're equally intolerant proteins in the body, and they'll probably accrue similar numbers of variants. So SCN 1A, 2A, and 8A, I think, predict the future of where GRIN's going to land. Of the 1,000 or so we know about, I stopped counting them. There's so many. I used to be able to keep track of everything. 500 are actually done. 415 are on the portal. We have the portal has an informal peer review with individuals outside of the lab that generates the data to look at the data and give a critical eye that it's acceptable or it's not. So there's actually another 75 done that we just haven't had a chance to push through our quality control pipeline, get to our reviewers, and get on the portal. So we're about halfway there, about 500. There's another 300 that we have partial data set for. And so, my optimistic, hopeful answer is for the GRINs, yes, it's feasible. That was the goal in 2011 when we started. Hongjae, Yuan, and I, when we first started realizing this was common, we sort of thought, all of them need to be done. And a model is CFTR, cystic fibrosis. All the variants are known. A new patient gets diagnosed, it's a lookup table. That's our goal with the variants, AMPA, NMDA, kainate, and GRIDS, is to get it all done and put ourselves out of business so that the clinicians can operate on that data. And as Ian said, we just need the time, the resources to execute the assays. We need to work together to, make sure we agree as a field pragmatically on what should be done. We picked 6 assays. We do 12. We picked 6. We thought, you know, is a compromise to allow us to do everybody and basically get the call right most of the time. We're on the edge of adding a 7th this year. Lonnie's working on an 8th that might get added. Tim Benke's working on a couple more in the C-terminal, so is Catherine Roche. But our goal is to get them all done. We're going to get them all done, and the field's going to move forward eventually.
Incredible. That's a lot of variants that have been characterized or in progress. That's amazing. Ian, do you want to comment on the GRIA genes at all?
Yeah, well, just, I mean, first checking back to what Steve just said, my heart sinks when I think about adding another couple of tests, particularly if you then need to go and redo, because these tests are likely to be things you could have done at the time if you'd have realized they were coming. So I hope that it's not something you have to repeat on all of the variants that have already been. In terms of the AMPA receptors, they are simpler in a way, in that so one of the things we've spent a lot of time over the last 2 or 3 years is trying to activate AMPA receptors in the kind of the way they are at the synapse, which is very, very quick, very, very short. So it's a different main technique than the one that Steve uses at Emory. And when you do that, you do see little subtleties in the loss and gain. You know, you do see there are several different kinds of loss of function, and there are several different kinds of gain of function. But having spent all the time doing that, it's very frustrating that if you just get an oocyte pumped full of AMPA receptors and wash glutamate on it, almost all of the ones you'd think would be gain of function have an increased potency, and almost all the ones you think would be loss of function have a reduced potency. And so potentially you could be in a position where just a single assay on an oocyte actually covers you for the majority of functional changes that the AMPA receptors have. Now, I don't want to categorically say we'll be in that position once we finished our work, but nonetheless, if they are gain of function because they have a high affinity but desensitization is still intact, they have increased glutamate potency. If they have reduced desensitization and slowed deactivation, they also have increased glutamate potency. So it potentially can boil down to a much simpler field, a much simpler situation with the AMPA receptors. Yes, you have the complexity of more combinations of AMPA receptors, but in terms of the actual experiment you need to do, potentially it could be more straightforward. But we're not in a position. We're not going to put the Hexel amplifier in the box and put it on the high shelf just yet. But in terms of where we are in terms of GRIA variants, so I'm working in strong communication with Alan Byatt, who has a database. So he's an epileptologist, pediatric neurologist in Copenhagen, and he has over 200 GRIA patients on a database, and we are not that far through characterizing those, but equally, because with organizations like CureGrim, in general, the most kind of engaged, the most interactive parents tend to be in all of the databases, and often some of these variants that are in his database are already in published data sets. In terms of how long it's going to take us to get through the ones that currently exist, I still think we'd be probably several years away. Not that the experiments are necessarily that hard to do, but again, there's just nobody to do them. I'm expanding using students actually to kind of train them and get them in the lab, and that's gone well this year. We had just 6 brand new students who'd never patched anything on oocytes, and we got 11 variants through, and that will be scaling up. But everything that they do, I then need to go and reproduce, because I can't, it just is, there's a general, a kind of a due diligence that needs to be done with all of these bits of information before they can find their way back to the clinicians. So it's a long drawn out process, and it's not like GRIA disorders going everywhere. The variants are going to keep on cropping up. Like Steve said, there is likely to be almost a theoretical ceiling, whereby in terms of the spelling mistake variants, each amino acid will typically have about 7 different single nucleotide variants that can change the amino acid. And there's a limited set of amino acids within a protein which will be sensitive to variation. And eventually you are going to get in a position where every pathogenic variant there can be has already appeared before and been tested. But that is much further down the line, certainly for the rate we're going with GRI, I would say. Yeah.
Thank you. And so I'm going to ask this next question kind of touches on 2 points, which is, can you all very high level walk us through, how variant characterization is done or classification is done? For example, are you using brain cells, other cells, animals? Like what actually needs to be done? And with that, are there certain variants that can and cannot be tested? That's a question that we get a lot from families is, can my child's variant be tested? And how do you figure that out? Let's start with Lonnie this time.
Yeah, so we actually look at variants at 3 levels. But the most high throughput way for us is just expressing the variant in what's called a heterologous expression. We're expressing it in a very artificial system called HEC293 cells. They're just a cell that take the gene and turn it into a protein and put it on the membrane, and we can characterize those properties. In another system that Ian and Steve use is similar to that. It's called oocytes. So again, these are fairly artificial systems because you are, they're not neurons, they're not real cells, and but they have an advantage of one, you can control the composition of the protein. You're expressing the specific variant that you want. You can rapidly in a relative sense, characterize its functional properties, the way it works, how it carries current. And that allows you to do a large number. So I think that's sort of common amongst all 3 of us, is that we're going to use a system where we can express the protein and look at functional properties, this ability of the channel to carry ions across its membrane. Now, for our group, we go 2 steps further. One is, but these are much lower throughput now. One is human embryonic cells and iPSC cells. We're looking at the variants there. Again, though, these are now, we're focusing on a smaller number of variants because of costs and time and how much effort it takes. And then the third system we're using in collaboration with Dr. Howard Sorotkin is to look at these variants in zebrafish. And but again, both zebrafish and human embryonic stem cells, they offer a lot of advantages. They're real cells. You can assay behaviors, but like the rodent models, they take time to generate, and they're more expensive. Although zebrafish are much less expensive, are cheaper than rodents. So that's one of the major reasons we're very interested in this approach.
That's
but
great.
in terms of characterizing loss and gain of function, it's mainly using these artificial cells because of the speed at which you can do it. That you can look at a lot of variants per day, 3 to 5 to 6 a day.
Wow, okay.
Using that.
And can you characterize, so we talk a lot about missense variants, particularly in the, relationship to GRI genes. Can you talk a little bit about like larger deletions, smaller deletions? There was a conversation in a previous session about frame shift mutations. Like, can these other types of variants be functionally classified, and why or why not? Maybe you can just add on to that, and then we'll go to the other panelists.
Yeah, my group mainly focuses on missense mutations or missense variants. Those are the ones that they change an individual amino acid in the protein, and we characterize it. We do look at some truncations in the C-terminal domain, but other truncation, nonsense mutations, depending on where they occur, if they're very early in the gene or in the protein's not going to get made, and that's going to create things that really depend on the cell biology, with the haploinsufficiency, etc. So we really focus mainly on truncations in the C-terminal domain because the protein is still there. It's just lost part of that intracellular part of the protein that's there. And mainly missense mutations.
Thank you. Steve, do you want to add on anything?
Sure. So, I guess a couple things up front. We look at the function two ways. We have academic programs, funded intermittently by the NIH to make animals, and we look at microcircuit and behavioral effects, and those are really always aimed at corrective actions, precision pharmacology, as well as genetic therapies. And so we've made four or five mouse lines at Emory. We collaborated with Wayne Frankel to make another 3, and we've brought a couple in from the NIH. And that's an important way to look at function. And we did that early on with our strongest variants identified in heterologous systems that Lonnie nicely described, and convinced ourselves that the same properties that we saw shifted in HEC cells and oocytes were shifted in neurons. And so that gave us some confidence that we're not wasting our time in the heterologous systems. So that's one functional assessment. In terms of trying to make calls for the clinicians, we started with 12 parameters that we've measured on and off, and our goal all along was to do all of them and do them in a comprehensive fashion. And so, pragmatically, we had to make decisions. iPS cells are great, but they take many, many weeks to grow. They're very expensive, 30 times the cost, maybe more, the cost of oocytes or HEC cells. It's absolutely not feasible to do 500 variants. For me, with current technology, we're talking 10 or 20 variants in that system. So we avoided that system, and we stuck with the systems Lonnie described, the HEC cells and the oocytes on our screening platform. And most of our time, once we settled in on the 6 assays, and Ian, we do have a trick for the 7th assay that we're going to go back. We think we can squeeze some data out of a set of recordings. I'd be happy to know. But those 6 and possibly 7 assays we have refined. We're now working on this for 15 years. We've set up an automated pipeline. Right now, we're undergoing a lot of federally controlled CLIA, what's called CLIA compliance in the U.S., where every item in the lab is certified yearly, and there's a very tight inventory and a quality management system like you would have in industry. And we have robots, and we have automated pipelines where data gets recorded and analyzed overnight and formatted and spit out for managers to sign off. We've sort of set that up with those 6 assays, and the 6 assays are the potency of the activating agent, glutamate and glycine, the potency of a main inhibitory element, magnesium, just an ion in your brain that shuts down the channel, and as Lonnie said, gives it all kinds of really interesting properties that allow it to do its job in biology. We look at the time course of response, a little bit like what Ian was describing with the AMPA receptors. And then we look at its surface localization, and then we have a trick to ask, what's the theoretical probability a receptor that binds an agonist will actually open? So those are our 6 assays. And they all run through this pipeline. We now source the DNA from a third party with validated documentation, and they move through, and the goal is, if FDA knocks on my door tomorrow and says they're going to audit our laboratory, everything is perfect so that data can be used to make clinical decisions. For example, patient selection in a trial. So we divide function into those. We think of it those 2 ways. The academic ones, animals trying to conceptualize and innovate on rescue pharmacology and validate the heterologous systems, and then this heterologous pipeline where we're trying to be as careful as possible and gear up to generate data that can be used by clinicians.
Great. And just for the sake of time, I'm going to now switch gears a little bit, and we're going to focus on specific questions that are coming up in the chat. If any of the panelists want to reply directly to any of these, please feel free. But one thing I want to make sure gets addressed is, how can families looking to get their variant classified go about doing that? who should they reach out to? Who can they contact? Whoever wants to take that one, just jump in.
Maybe I'll start. I used to encourage parents to contact me, and I'd rather interact with parents than Emory paperwork and compliance and things I'm late on and bureaucracy. We're actually at a place now that the federal government's involved in regulating what we do so it can be used for clinical trials, where I'm legally no longer at CFIRV allowed to interact with parents. And so you can contact me, and what I will do is, if you're in the United States or Canada, I think I'll pass your contact on to a free agency called Informed DNA that will give free consulting advice and make you aware of any clinical trials ongoing. And I will also at the same time pass you on to Tim Benke, who runs a registry. I'm a firm believer that every parent should be in one of the registries. I think it will catalyze understanding the disease, and also really helps the companies know that they can go and get clinical phenotypes that they need for figuring out whether they're going to pursue an indication or not. If you're from Europe, then I pass you on to Johannes Lemke. There is no equivalent of Informed DNA in Europe, and all the different privacy rules in the country are slightly different. And so there isn't a unified framework, but Dr. Lemke is connected, at least in the context of clinical trials, to virtually all of the sites in Europe and interacts with them routinely. If you want your variant characterized, then you can contact any of those directly, but if you contact me, a polite email will come and we'll move it out, and that's simply because the regulators said, and the lawyers said, now that you're doing human diagnostics and clinical trials involved, you're no longer allowed to interact with patients, because that breaks de-identified nature. When a DNA comes to our lab, I have no idea who it's from or anything. But the moment the parent says, "Hey, my daughter has this variant, can you work on it?" Suddenly it's no longer de-identified, and I'm running across federal rules.
Thank you. Ian, can you maybe talk very briefly about how to do this for GRIA variants?
Yeah,
so for other genes.
Yeah, so I have a data sharing agreement with Alan Byatt for his database. So if you contact him, he will then, for the same reasons that Steve just went through, it's kind of interesting that, so much of my interaction with CureGrin over the last few years, it seems to be in pretty much direct contradiction with an ethical agreement UCL has just made me sign as part of, so I would imagine that a conversation I have in a conference it's fine, but as soon as there's any kind of paper trail involved in terms of emails and and occasionally just out the blue, someone will email you their full genetics report and the data protection stuff, and it's a bit of a mess. So yeah, so going to Alan Byatt is fine. I've also at the moment I'm going through the process of signing another data sharing agreement with Kristen Park. So I think if you do go to Tim Benke with your GRIA, there is the chance now, if they want me to, they can potentially share that information with me as well. So again, this is multiple different routes. I don't think there's any problem with signing up for more than one database. I mean, I think the information is kind of useful. I suppose if we're not very organized, we might think, oh wow, look, there's 3 patients with this variant, we're going to work on it. It just so happens you've signed up to all of the registries, and so that I suppose in that regard, maybe that's something you want to consider. But yeah, so Alan Byatt and Tim Benke are the ways in for GRIA, much the same as it is with Tim and Johannes for GRIA.
Thank you. Can one of you maybe answer this question in the chat about how should we go about interpreting conflicting functional analysis when variants exhibit both loss of function and gain of function parameters? And what does that actually look like on the GRIN portal as well? That might be good to mention. Whoever would like to take it.
I'm happy to take that. Maybe Ian and Lonnie can chime in and clarify it. So there's, we measure 6 parameters, and I would say nearly 2/3 of the time, there's a pretty clear result. And a third of the time, one parameter will enhance the function, another parameter will decrease the function, and sometimes you got 3 and 3 opposing each other. And so we have worked on the question of what to do with that numerous times, and the same question will come up with AMPAs, and and the same question will come up with the kinates that are out there sometimes. And so our strategy was the following. We're working with 2 different biostatistician groups on a Bayesian type system, but it's not really helpful. And what we fell back on was a pseudo-Bayesian system where we treated each parameter independently, and we came up with an average contribution, but not where everybody is weighted equally. Everybody is weighted by the math by which they contribute to a synaptic or non-synaptic current. And that's where it differs from a Bayesian analysis. And so a glutamate potency change is viewed through the eyes of the concentration actually in the cleft and how long it's in the cleft and will it bind or won't it bind. And a time course change is viewed through the eyes of will it change a synaptic current, and would it change a non-synaptic current that was activated by basal glutamate outside of a synapse. And we found when we weighted things that way, we can use that as our tiebreaker, with statistical thresholds for times when we see parameters going in both directions. And we liked that tiebreaker because we've analyzed, I'm not sure how we got to this place, but we've analyzed almost 200 variants in healthy individuals for 3 or 4 parameters. These are patients in the NOMAD database. I mean, we started this in 2011 when we were first trying to figure out what are the thresholds for a change, how much variation there is there normally, and then when do you go beyond that as potentially pathogenic. But now those patients there helped set all these thresholds for the tiebreakers and when we're going to call a change meaningful. And that tiebreaker resolves us, maybe half the time, and then we're left with 15% where the receptor does not work normally, but we can't squeeze you into this simplistic view. Lonnie nicely said, early, very crude first step categorization of gain or loss. And then there isn't a gain or loss. It's just the receptor's different. It's complicated. The data needs to be there because down the road, smart people will look at it and eventually make headway.
Thank you so much. We've reached the end of the panel, but I just want to thank all of our speakers, Dr. Wollmuth, Dr. Traynelis, and Dr. Coombs. This has been fantastic. So thank you so much for sharing your insights. And for everyone else, we can head back to the main room for closing sessions, or for the closing session. Thank you.
Thank you, Quinn, for
Okay,
organizing.
thanks very much.
Thank you very much.
Thank you.